Good evening, welcome back. We're on to the continuation of the lessons on DNA replication in chemistry. So this is basically biochemistry. So this is a picture of the human mitochondrial DNA with 16,569 base pairs. The explanation below states that the map of a human DNA where the genome encodes for 13 mRNA green, 22 tRNA violet. So that's, you can just have a look at the picture here. See this? Right? So I'm just magnifying it so you just get to read the details. And it has about 2 RNA pale blue molecules. There is also a major non-coding region NCR, which is shown enlarged at the top in blue. The major NCR contains the heavy strand promoter HSP, the light strand promoter LSP, 3 conserved sequence boxes CSP 1 to 3 orange, and edge strand region of replication OH of the termination associate sequence TAS yellow. The triple stranded displacement loop D-loop structure is formed by premature termination of nascent edge strand DNA synthesis at TAS. The short edge strand replication product formed in this manner is termed 7S DNA. A minor NCR located approximately 11,000 base pairs downstream of OH contains the L strand, origin of replication OL. Now, a dedicated DNA replication machinery is required for its maintenance. Mammalian mitochondrial DNA is replicated by proteins distinct from those used for nuclear DNA replication, and many are related to replication factors identified in bacteriophages. DNA polymerase Y, poly-Y, is the replicated polymer in mitochondria. In human cells, poly-Y is a heterotrimer with 1 catalytic subunit poly-A, pol-YA, and 2 accessory units pol-YB. So pol-YA belongs to the A family of DNA polymerases and contains a 3' 5' exonuclease domain that acts to proofread the newly synthesized DNA strand. Pol-Y is a highly accurate DNA polymerase with a frequency of misincorporation lower than 1 × 10⁻⁶. The accessory pol-YB subunit enhances interactions with the DNA template and increases both the catalytic activity and the processivity of pol-YA. The DNA helicase twinkle travels in front of pol-Y, unwinding the double-stranded DNA template. The twinkle forms a hexamer and requires a fork structure, a single-stranded 5' DNA loading site, and a short 3' tail to load and initiate unwinding. Mitochondrial single-stranded DNA binding protein mitochondrial SSB binds to the former ssDNA, protects it against nucleases, and prevents secondary structure formation. The most accepted model of DNA replication in the mitochondria is the strand displacement model, which is shown in figure 9.24. Within this model, DNA synthesis is continuous as both the H and L strand. There is a dedicated origin for each strand, OH and OL. First replication is initiated at OH and DNA synthesis then proceeds to produce a new H strand. During the initial phase, there is no simultaneous L strand synthesis, and mitochondrial SSB covers the displaced parental H strand by binding to single-stranded DNA. Mitochondrial SSB prevents the mitochondrial RNA polymerase pol-RMT from initiating random RNA synthesis on the displaced strand. When the replication fork has progressed about 2/3 of the genome, it passes the second origin of replication OL. When exposed in its single-stranded conformation, the parental H strand at OL folds into a stem loop structure. The stem efficiently blocks a mitochondrial SSB from binding, and a short stretch of single-stranded DNA in the loop region therefore remains accessible, allowing pol-RMT to initiate RNA synthesis. Pol-RMT is not a processive pol-RMT is not processive on single-stranded DNA templates. After adding approximately 25 nucleotides, it is replaced by pol-Y and L-strand DNA synthesis is initiated. From this point, H and L strand synthesis proceeds continuously until the two strands have reached full circle. Replication of the two strands is linked since H strand synthesis is required for initiation of L strand synthesis. DNA ligase 3 is used to complete the ligation of the newly formed DNA strands. During DNA replication, the parental molecule remains intact, which poses a steric problem for the moving replication machinery. Topoisomerases belonging to the type 1 family can relieve torsional strain formed this way by allowing one of the strands to pass through the other. In mammalian mitochondria, top 1MT, a type 1b enzyme, can act as a DNA swabber, working together with the mitochondrial ribosome. Furthermore, replication of circular DNA often causes the formation of catenanes or interlocked circles that need to be separated from one another. The type 1 topoisomerase 3-alpha, top 3-alpha, is required to resolve the hemicatenane structure that can form during mitochondrial DNA replication. Please take screenshots as we keep proceeding. Right, and I'll just bring it down again for you, just in case you need a screenshot there, right? And just move it up there so you can get a good picture of the whole thing, right? And now we're going to the explanation on that. Replication of the human mitochondrial genome. Mitochondrial DNA replication is initiated at OH and proceeds unidirectionally to produce the full-length nascent H strand. Mitochondrial SSB binds and protects the exposed parental H strand. When the ribosome passes OL, a stem loop structure is formed that blocks mitochondrial SSB binding, presenting a single-stranded loop region from which the pol-RMT can initiate primer synthesis. The transition to L-strand DNA synthesis takes place after about 25 NT when pol-Y replaces pol-RMT at the 3' end of the primer. Synthesis of the two strand proceeds in a continuous manner until two full double-stranded DNA molecules have been formed. Curiously, not all replication events initiated at OH continue to full circle. Instead, 95% are terminated after about the first 650 nucleotides at a sequence known as the termination-associated sequences test. This creates a short DNA fragment known as the 7S DNA that remains bound to the parental L strand, while the parental H strand is displaced. As a result, a triple-stranded displacement loop structure, D-loop, is formed. The functional importance of the D-loop structure is unclear, and how replication is terminated at the TAS is also not known. The next topic will be telomeres and replicative senescence. The end replication problem. In humans, telomeres consist of hundreds to thousands of repetitive sequences of TTA, GGG, that is thymine, adenine, guanine at chromosomal ends for maintaining genomic integrity. Because the DNA replication is asymmetric along double strands, RNA primer sequence at the 3' hydroxyl end cannot be replaced by DNA polymerase 1, as there is no 3' OH primer group present to the polymerase for the polymerase to extend the DNA chain. This causes the loss of 300 to 200 nucleotides with each DNA replication in cell division and is known as the end replication problem. Telomeres provide a repetitive non-coding sequence of DNA at the 3' end to prevent the loss of critical genetically encoded information during replication. Moreover, telomeres are coated with a complex of 6 capping proteins, also known as shorten proteins, which are packed into a compact T-loop structure that hides the ends of the chromosomes. This prevents the DNA repair machinery from mistaking chromosomal ends for double-stranded DNA breaks. Therefore, telomeres have been proposed as a mitotic clock that measures how many times a cell has divided and in essence gives a cell a defined lifetime. Note these diagrams. Take screenshots, please. right? I'll just bring it down here, right? This side, and I'm moving it slowly so that when you make it a still screen, you can always go for good screenshots. Now the telomere structure, telomeres are located at the end of chromosomes where they help protect against the loss of DNA replication. B. DNA quadruplex formed by telomere repeats. The looped conformation of the DNA backbone is very different from the typical DNA helix. This is known as T-loop formation. The green spheres in the center represent potassium ions. Have a look at the green spheres at the center are the potassium ions, right? Now the human telomerase enzyme is responsible for maintaining and elongating telomeres and consists of RNA component TERK and reverse transcriptase TERT that serves as the catalytic component. The TERT uses the TERK as a template to synthesize now new telomeric DNA repeats at a single-stranded overhang to maintain telomere length. Some cells, such as germ cells, stem cells, hematopoietic progenitor cells, exuberant lymphocytes, and most cancer cells, constitutively express telomerase and maintain telomerase activity to overcome telomere shortening and cellular senescence. However, most of the somatic cells generally have a low or undetectable level of telomerase activity and concomitantly limited longevity. Interestingly, overall telomerase activity decreases with age, but increases markedly in response to injury, suggesting a role for telomerase in cellular regeneration during wound healing. The telomerase length and integrity are regulated through the interplay between telomerase and shorten proteins. Please take a screenshot of these diagrams, right? Okay. Conceptual model of telomerase activity. The active site of the telomerase enzyme contains the RNA template, TERK, shown in red, and aligns with the last few telomeric bases at the end of the chromosome, shown in blue. This creates a single-stranded overhang that can be used as a template by the TERT reverse transcriptase to extend the telomere sequence. Check out the TERT and TERK, shown in red, TERK shown in red, and TERT in blue. See this, right? The active sites are telomerase. The RNA template telomerase ERC and template by TERT reverse transcriptase. The telomerase base at the end of the chromosome shown in blue. See, and the TERK there. In vivo shortened telomeres and damaged telomeres generally caused by reactive oxygen species ROS are usually assumed to be the main markers of cellular aging and are thought to be the main cause of replicative senescence. In vitro, telomeres lose approximately 5 to 200 base pairs at each division due to the end replication problem. Approximately 100 mitoses are thought to be sufficient to reach the Hayflick limit, or the maximum number of mitotic events allowed prior to entering replicative senescence. Right? So remember, what's the Hayflick limit? Is the maximum number of mitotic events allowed prior to entering replicative senescence. Cells in continual renewal, such as blood cells, compensate for telomere erosion by expressing telomerase. The only enzyme able to polymerize telomeric sequence de novo at the extremity of telomeres. Knocking out telomerase components such as the catalytic subunit TERT or the RNA template TERK induces several features of aging in mice. In humans, germline mutations and telomerase subunits are responsible for progeroid syndromes such as dyskeratosis congenita, a rare genetic form of bone marrow failure. Furthermore, healthy lifestyle in humans is positively correlated with the longer telomere length, and patients suffering from age-related disease and premature aging have shorter telomeres compared with healthy individuals. An accumulation of unrepaired damage within telomeric regions has also been shown to accumulate in aging mice and non-human primates, suggesting the damage of telomeres with age may also be a contributing factor to age-driven disease states and reduced health span. Thus, one could argue that the activation and expression of telomeres may be a way of reducing age-related diseases and increasing overall longevity. However, the constitutive expression of telomeres, unfortunately, is a characteristic of almost all cancer cells. It is therefore no surprise that transgenic animals overexpressing the catalytic subunit of telomerase M-TERT develop cancers early in life. However, overexpression of telomerase in mice that are highly resistant to cancers has shown large errors in median lifespan and significant reduced age-related associated disorders. Since humans are not highly resistant to cancer, this is not a feasible option for humans. However, additional studies in mice where constitutive expression of telomerase is only introduced into a small percentage of host cells using adenovirus gene therapy techniques has yielded more promising results. Adenoviruses are a group of viruses that form an icosahedral protein capsule that houses a linear double-stranded DNA genome. Infections in humans typically causes symptoms of the common cold and are usually mild in nature. These are a good target for gene therapy as the DNA that they carry can be mutated so that they are deficient in their ability to replicate once they have infected the host. They can also be transferred to carry a gene of interest to the host, but the gene can then integrate into the host genome. Experiments in mice that were infected with an adenovirus carrying the M-TERT gene showed that M-TERT was delivered to a wide range of tissues within the body and increased telomere length with those tissues. Furthermore, the M-TERT expressing mice were healthier than their litter mates and displayed a reduction in disabling conditions associated with physiological aging, such as osteoporosis and insulin resistance. Cognitive skills and metabolic functions were also improved. Noticeably, mice treated with gene therapy did not have increased incidence in cancer rates, suggesting that in at least the short-lived mouse species that a gene therapy approach to increase telomere activity is safe. Within these animals, median lifespan has increased by 24% when animals were treated at 1 year of age and by 13% if treated at 2 years of age. This is a diagram of that. Please take screenshots. Promoting health span in mice using a telomere gene therapy. Delivery at the catalytic subunit of telomerase TERT using a adenovirus vector RAAV. Suppresses aging-associated telomerase erosion and extends short telomerase in a variety of tissues. Consequently, animals display improved health span and extended lifespan. Next is the replication and repair of tissue of telomere sequences, right? In addition to the end replication problem, telomeric DNA, tel DNA replication and repair is a real challenge due to the different structural features of telomeres. Firstly, the nucleotide sequence itself consists of an hexanucleotidic motif, 2TA-G, repeated over kilobases with the 5' 3' strand named the G strand due to its high content in guanine. During the progression of the replication fork, the lagging strand corresponding to the G strand forms G quadruplex G4 structure, which have to be resolved to allow fork progression and to complete replication. Secondly, R loops corresponding to highly stable RNA DNA hybrids involving the long non-coding telomeric transcript telomeric repeat containing RNA also have to be dissociated. Thirdly, the extremity of telomeres adopts a specific loop structure, the T loop, which has to be unraveled. This is the loop that hides the double strand end from the DNA damage sensors and is locked by the hybridization of its 3' single strand overhang extremity with the above 3' 5' strand, thereby displacing the corresponding 5' 3' strand to form a D-loop displacement loop structure. Lastly, replication also has to deal with barriers encountered elsewhere in the genome, such as torsions and a condensed heterochromatic environment. Note these diagrams, they could be very helpful. So the figure 928, obstacles and solutions to replicate telomeres. Telomeric sequence with the G strand in solid red line and the C strand in solid green line is depicted. That is what you see here, right? Green strand and C strand is in solid green, G strand in solid red. All right? And freeze the parental G strand, enabling the formation of G4 secondary structure. R loops corresponding to Tera hybridization in dotted black lines. Let's see that. Dotted black lines. The Tera. Right? With the 3' 5' strand and torsions due to the fork progression are also known. B. Replication helpers such as helicases, either helping in G4 unwinding or in D-loop unlocking, are depicted. The DNases, top 2A, DNA2, and RNases. RNase H1 and FEN1 help in resolving torsions and RNA DNA heteroduplexes, while timeless stimulates the replosome, and POT1 competes with the RPA1 for binding of the single strand and helps in G4 dissolution. The shorten components, POT1, TRF1, and TRF2 help in loading the helper proteins, which are shown by the fine green arrows. Let's have a look at that. Where are the fine green arrows? There you see. And now, since telomeres face a host of obstacles to completing the replication process, as discussed in figure 9.28, the cell possesses a set of specialized machinery to fully achieve the replication, such as the RGE-L1, TRF1 and TRF2 proteins, DNases, RNases, and timeless. The recruitment of these factors is orchestrated by the shorten complex. At the molecular level, the GGG telomeric repeats are particularly sensitive to ROS, which produce stretches of 8-oxoguanine that are especially difficult to repair. Coupled with inefficient telomere repair, these ROS-induced lesions produce single and double strand breaks and or generate replicative stress, ultimately resulting in telomere shortening. The presence of unrepaired single or tandem 8-oxoguanine drastically inhibits the binding of TRF1 and TRF2 and impairs recruitment of telomerase, especially when ROS damage is localized in the pre-3' overhang. This type of damage contributes to telomere deprotection and shortening. Strikingly, ROS and other metabolic stresses also induce the relocation of TERT to mitochondria, as observed 1. in primary neurons after oxidative stress, 2. in neurons exposed to the tau protein, 3. in Purkinje neurons subjected to the excitotoxicity, and 4. in cancer cell lines treated with G4 ligand. Mitochondrial TERT increases the inner membrane potential as well as the mitochondrial DNA copy number and decreases ROS production with a protective effect on modern mitochondrial DNA. Mitochondria are also critical sensors of cellular damage and contribute to the processes of autophagy and apoptosis, programmed cell death. The relocalization of TERT following chromosomal damage in the nucleus may indicate one mechanism. The mitochondrium utilizes to monitor cellular stress and damage. So now we're going on to master the contents. Which of the following is the enzyme that replaces RNA nucleotides and a primer of DNA nucleotides? These are all questions I'm giving you. You can just note down the questions, take screenshots, answer them, and check the answers. There are answers here, right? Q, show answer, reveal answer, all these things that you figure it out. So the answer B, DNA polymerase is the enzyme that replaces the RNA nucleotides in a primer with DNA nucleotide. Which of the following is not involved in initiation of replication? The answer is A, ligase is not involved in the initiation of replication. Answer. Which of the following enzymes replication is unique to eukaryotes? That would be telomerase is unique to eukaryotes, right? And which of the following would be synthesized using this sequence as a template? It is 3 prime. We'll have to right check, get that. Answer: the enzyme responsible for relaxing supercalled DNA to allow the initiation of replication is called DNA agarase or topoisomerase. They unwind, right? Unidirectional replication of circular DNA molecule like plasmid that involves nicking one DNA strand and displacing it while synthesizing a new one is called rolling circle replication, right? Why is a primase required for DNA replication? What is the role of single-stranded DNA, protein DNA replication? So below is a DNA sequence. That's schema 3. Envision that this is a section of a DNA molecule that is separated in preparation for replication. So you are only seeing one DNA strand construct the complementary DNA sequence. Review figure 1 and figure 2. Why was it important that Messelson and Stahl continue the experiment to at least 2 rounds of replication after isotopic labeling of the starting DNA with 15N, instead of stopping the experiment after only 1 round of replication? If deoxyribonucleotides that lack the 3 OH groups are added during the replication process, what do you expect will occur? So these are the references, right? They're pretty helpful for in biochemistry and principles of biochemistry, cell metabolism, case prokaryotes, and similar ones. Okay, these are all the items. That will be the end of this lecture. We're stopping shortly, and just note these in your sections and take screenshots, and you could read more. And we have more coming up with chapters in biochemistry 1 and 2, and foundations of biochemistry, protein structure, etc. Will coming up soon, right?